Hi, everyone. Got a little bit of echo, so I apologize.
We might want that. We might
Cords
not
are
need
short. So excited to see everyone here and on Zoom. This is our first presentation in person in over 3 years. So I'm just really excited that we've been able to do this and that you've all come out to see us. I want to acknowledge and thank the Legacy and the crew for giving us this opportunity to hold the in-person meetings here and for spending their evenings with us and after being out on the water all day. We're very grateful to you, and Quinn is the only one here tonight, so I want to give Quinn a big thank you. Also, for those of you who could not make it tonight, we're pleased that you're joining us from home or wherever you are on Zoom, and we hope that you can see okay. I know there's a lot of glare. There we go. Yeah. So I want to announce that we have a field trip this Saturday, June 17th at 10 a.m. at the Hubbs-SeaWorld Research Institute hatchery in Carlsbad. There is parking, but we ask that you carpool because the parking is limited, and there is additional parking on the street at the entrance. There's no dress code, but it is going to be indoor and outdoor, and it's supposed to be a pretty warm day. I think 77 was the high as of today. It could, obviously it will probably be different on that day. For the address and more information, check our website at acssandiego.org. Our July speaker, July 12th, is Jessie Krantz. She's from the Alaska Fisheries Science Center's Marine Mammal Laboratory, and she'll give us updates on saving the North Pacific right whale. The presentation will be here on the Legacy in person and on Zoom, and that will be at our regular time at 7 p.m. Yes, she's coming to Alaska to spend time with us. She went to school here and was excited to have the opportunity to come back home. So we're excited to have her here on the Legacy. Tonight, we're going to have an opportunity drawing after the presentation. So please hold your questions until after the presentation. And of course, for those of you on Zoom, please put any questions in the San Diego chat room or chat area. So our speaker this evening is Danielle Haulsee, and I'm so glad you're here tonight. About 2 weeks ago, she emailed and said that she had to be in DC for meetings tomorrow and Friday. And selfishly, I thought, holy crap, it's our first in-person presentation, but she's a trooper and she is foregoing her sleep and decided to take the red eye out tonight so that she could be here with us today. So we're so happy that you're here. Danielle is the Chief Science Officer at Hubbs-SeaWorld Research Institute and specializes in fisheries, oceanography, and the spatial and behavioral ecology of marine animals. She'll tell you more, but she gets to work with robots too. I think that's kind of cool. Danielle earned her Ph.D. from the University of Delaware's College of Earth, Ocean, and Environment, focusing on characterizing the movement, ecology, and social dynamics of the sand tiger shark, a coastal top predator found along the east coast of the U.S. And I think they had no spottings around here. Never mind.
Not on this.
I know we've had shark sightings, but apparently not tiger shark sightings. Just like ACS, Danielle is passionate about education and is a promoter and mentor of STEM to the underrepresented groups through her participation in organizations like the Society for Women in Marine Science, the Gills Club, the Sky Scientist Program, University of Delaware's Mentor Collective Program, as well as serving as a mentor for many other high school and undergraduate student internship opportunities. Without further ado, Tags and Tech to Support Conservation and Management in a Dynamic Ocean by Danielle Haulsee. Thank you.
Thank you so much for having me, and I'm excited to be here. I'm gonna try to remember to stand right here because it blocks this sun that's coming through, but if I move, I apologize to those on Zoom. So that was a great introduction, and I appreciate the introduction because I'm gonna be sharing with you all kind of a bunch of different projects that I've worked on throughout my past, including the sand tiger shark, so you get to see what those guys look like that are found on the East Coast. But focusing on how we use different kinds of tags and different kinds of technology to support conservation and management. Oh. All right, so, and there's also, I changed the slide and when it shows up for you guys on here, so I'm gonna try to remember that, but if I start talking about something and it doesn't make sense as to what's on the slide, that's why, because I see there we go. Okay, so I'm gonna actually start by having you guys think about how we manage and conserve our resources on land. So take, for example, the millions of amazing wildebeest and zebra that migrate across the Serengeti. This is a natural phenomenon that I think we are all aware of. We see it in documentaries all the time. And the importance of this natural wonder has not been lost on researchers who have studied this natural wonder in really great detail. And when we have something so amazing that we want to conserve and protect on land, often our solution is to draw a box around it. So here in this map that hopefully you can see is a simplified rendering of what the annual migration is. And you can see the boundaries are the Serengeti National Park and several other conservation and management and game reserve areas that we've kind of drawn as the box around this phenomenon. And in this way, we can put up fences, we can limit access to these resources, we can use visual population surveys to see how the populations are doing and try to identify threats to to this natural wonder. And so in this way, we can draw a box around it and protect and conserve. Now, in the ocean, I'm going to argue that there are similar mass migrations that are happening, but it's just harder for the rest of us to see. So, for example, along the east coast of the United States in the Atlantic Ocean, millions of bony fish and elasmobranchs like these sand tigers that you see here are taking part in these really amazing annual migrations where they're spanning entire coastlines and millions of available ideal habitats and food resources. But the problem is that it's a lot harder for us to see this natural wonder, and it's a lot harder for us to figure out how do we protect and conserve these resources or these things that we care about. So one example that we've tried to take kind of what we're doing in the terrestrial world and implement that in the ocean is the idea of drawing a box around it. So here on this map, it's very, I'm not sure why it's, yeah, very pixelated for you. You get it. Okay, so this is a map of the east coast of the US, and it's off of Virginia and North Carolina. The box that is drawn here is the Mid-Atlantic Shark Area Closure. So this is a time closure where managers have decided we're going to draw a box along this area for a certain time of the year. We're going to close off to fishing to protect sharks that are migrating through. Now the black dots that you see there, so all of the dots are locations of sand tiger sharks that were carrying tags. The black dots are the dots of sand tigers that traveled through the time area closure when it was in effect. So you can see hopefully you can see there's very few black dots. Most of the dots are white, which means that the time area closure was mismatched in space and time for when these sharks are traveling through this area, not only the sand tigers that we're trying to protect, but the other shark species that we're interested in protecting as well. And so where's this breakdown? Now, it's actually pretty simple. When we're comparing the ocean to land, we're comparing kind of a dynamic ecosystem to a relatively static ecosystem. You can't read this, but it says dynamic versus static-ish. So really when we're talking about habitats in the water versus on land. In land, everything is in constant flux because of the nature of life in water. Everything from the nutrient availability to the habitat types, the primary producers on up to the top predators, everything's in constant flux. Whereas on land, things are relatively static. So our primary producers, our grasses, grounded, and that helps ground the ecosystems for relatively larger spatial scales and longer temporal scales. And so one way that researchers are trying to combat this is to implement new strategies, new thinking on how we're managing our ocean resources through something called dynamic ocean management, which is an adaptive approach that uses near real-time data to inform the spatial and temporal distributions of management or conservation actions. And a really classic case study of this is actually occurring out in the California current off of our coast right now. So historically, there's existed a drift gillnet fishery, which was primarily targeting swordfish and thresher sharks but also had a ton of issues with catching other things. So bycatch, things that the fishers weren't intending to catch, things like turtles, dolphins, seabirds, oh my goodness, whales, things that you guys care about, as well as us. And this is a breakdown of just a few years of that fishery and the numbers of these protected species that were getting caught up in these drift kill nets that were trying to target things like swordfish and thresher sharks. And so this was a concern to managers. We don't want to be catching these protected resources. And so what we tried to do is kind of implement what we do on land, draw a bunch of boxes and create a bunch of time area closures. So on this map here, this is listing all of the different kinds of time area closures that are going on. This panel here shows you the time of year when they're in effect, and then you can see the spatial extent of those. You can see a large portion of the west coast of the US is made as closed at certain times of the year for the Pacific leatherback conservation area. And at various times of the year, there's multiple time area closures overlapping all over the place. It's really making it hard for the fishers to go out and also concentrating fishing effort in certain areas. All with good intentions, but it's having an impact, and that impact on the fisheries has economic consequences. So this next plot that'll show up will show you the decrease in landings and vessel permits for those fishers who were targeting a relatively sustainable stock of swordfish out in the Pacific Ocean. And you can see with just all of the constraints that we're placing on them to protect the protected species that we care about, it really has collapsed the fishery over the last couple of decades. And so because of this kind of conflict between there's some resources that we want to access, but there's resources that we also want to protect and conserve, this idea of dynamic ocean management came to be. And this was put forth by Sarah Maxwell and others in 2015, where there was kind of this hypothetical idea that if we had a better understanding of the kind of the fine scale distribution of these protected resources and also the resources that the fishers are trying to access, then we could hypothetically have time area closures that are more dynamic, more reflective of the dynamic nature of water. And so they actually implemented this into modeling efforts where they were modeling swordfish availability, sea turtle availability, pinnipeds, so sea lions, sharks, a couple different species of sharks, and they were able to produce these maps that were predictive of there's a high likelihood of these species being here in a very dynamic way. And these models are fed by satellite data that is, we're getting new satellite data every day. And so what they did is created a tool called EcoCast, which basically took all of those inputs for the different species and put them all together, and also included risk weightings. So for example, if we really, really care about limiting our bycatch of Pacific leatherbacks because they're critically endangered, then we can say that we are very much concerned about that, and that has a higher risk weighting. And so the output of this model is actually these maps which show areas that are better to fish in blue and poorer to fish in red. And so these are actually recommendations that can be updated on a daily level, fed to fishers, and then make a choice themselves to say, I don't want my fishery to get shut down because we're catching too many of this protected species. So I'm going to choose to go to areas where this model is predicting that it'll be better to fish and my chances of bycatch will be lower. And they actually found that if we were able to implement this kind of dynamic closure areas, it would actually open up 30 to 40% of fishing grounds back open to the fishers. We're going to talk more about that at the end. So that's kind of the classic case study of what dynamic ocean management is. It's an integration of biological, environmental, socioeconomic, and policy information, bringing that all together to create models and tools that we can then use to hopefully implement a more dynamic management of our ocean resources. And my research has often working in systems that aren't as well studied as the California current ecosystem, and so has focused a lot on just collecting the biological, environmental, and more recently integrating socioeconomic and resource user information to help create these tools. So I'm going to transport us over to the Atlantic Ocean now and talk about some of the work that I did on sand tiger sharks on the East Coast. So sand tiger sharks are an amazing coastal predator, but unfortunately they're not, white sharks or tiger sharks. They don't come up to the surface and fin, so we're not able to use things like GPS tags on them. We have to get a little bit more creative, and what we end up using very frequently for these species is something called acoustic telemetry. So acoustic telemetry in its simplest parts is made up of two things: an acoustic receiver that we deploy in the ocean somewhere on a mooring, and then an acoustic tag, which is either attached to the outside or internally implanted into the animal, carries the tag, and whenever it swims within the detection range of the receiver, the unique identification of that animal is picked up and logged on the receiver. So we know that shark XYZ was here on this day at this time. And then, so if we have enough receivers in the areas that we're trying to monitor, we can then kind of reconstruct their movements in that way. One important thing to note, though, is that all of that data is archived on this receiver. At least at the time when we were doing this study, they now have receivers that will have a satellite uplink. They have a cable that goes up to a surface, and then there's a satellite uplink, but they're still really expensive. So most of these receivers are archiving all of this data. And because we have to then go out and collect the receiver and download it, they're often concentrated in coastal areas that are easy to access by researchers, because we have to go out on a boat and haul them up and download all the data off of them. So this is an example of the receiver array that I was working with in the Delaware Bay, which was actually very extensive. There's hundreds at any given time. There were almost 100 receivers out there. You can see they're concentrated in the bay and then the coastal ocean, but there's still gaps, right? We're not able to monitor everywhere. And so one thing that we did was we used robots. So we worked with autonomous underwater vehicles. This is a glider. So what's cool about gliders is they don't have a propeller on the back of them to move. They actually they're buoyancy driven. So they will suck water in or push water out, and that changes their buoyancy. And then because they have wings on the sides, they will glide forward and then glide back up in this seesaw pattern. These instruments are really unique or really cool because they're also taking environmental measurements. So every few seconds, they're taking measurements of water temperature, salinity, oxygen, chlorophyll levels, so the productivity of the water, and then also measurements of water clarity. And what we did is we integrated those acoustic receivers into the top and the bottom of that glider so that we could also use that glider to track the animals that we were tagging with acoustic tags. So this is just a little cartoony example of what a glider mission looks like. You have the glider seesawing in the water, collecting these profiles of the oceanographic conditions, and then every few hours they come to the surface, they transmit that data back to us. That would include data on whether or not we detected a tagged animal, and then us, glider man on the computer, can send new instructions back to want or tell it to continue on its thing. And so we're able to create a mobile telemetry platform where we can start to listen for these tagged animals in areas where we didn't have receivers. And so this is, when the map comes up, is a track of one of the first glider missions that we ran with this integrated acoustic receiver technology in it. And so the black dots here, the gold line is where the robot actually flew in the mission. The blue dots are where we detected tagged sand tigers, and then these black dots are the receivers. And so you can really see how these moored receivers, they're not covering much of the ocean, but we're able to tell some things. But we were interested in this mission and using the robot to help fill in the gaps between these receivers and look for these tagged sharks while they were doing their fall migration south. So one of the other really useful and unique things about using a glider to collect some of this environmental data instead of, say, the satellites that are constantly going out there is the satellites only see the conditions at the first few centimeters of the water. But many animals are not up there at the surface. And like I mentioned about sand tigers, they're very rarely at the surface. Their name sand tigers comes from the fact that they're often associated with sandy environments near the bottom. And so really the conditions that are being measured by satellites at the surface might not be reflective of the conditions where they are down in the water column. And this is reflective, hopefully you can see here, this is the data from the glider. So all of those profiles from the glider going up and down, if you squish them all together, you get a plot like this. So the warm colors are its temperature, salinity, color dissolved organic matter, chlorophyll, oxygen, and then this line graph on the bottom here is how far from shore the glider was. And you can see at the beginning of the mission, we had a system where there was some stratification. So the waters at the surface were much warmer than the waters that were below. But then we had a fall storm come through and it mixed the water column up. And as this, now you can see the rest of the mission, conveniently for us, all of the conditions, the conditions at the surface were then reflective of the conditions at the bottom. What I'm showing you now with the white dashed lines are where the glider was. So what were the conditions when we detected one of those tagged sharks? So this is the first time we'd ever used a glider or a robot in this way to detect these types of tags. And so what we did is then matched up the conditions, the environmental conditions where we tagged sharks versus where we'd heard no sharks, and we looked for habitat selectivity. And what we found was that the conditions that these sharks were selecting for were waters that were lower in salinity, higher in color dissolved organic matter, and closer to shore. And this is of the Delaware Bay water mass as it's exiting the bay, and then it hugs the coast to the south. And so what we were really tracking is the association of the sand tigers with this water mass, that they were leaving the bay and then hugging the coast to the south during their fall migration. And so this was really informative as we wanted to scale up our predictive modeling of where sand tigers were in the coastal ocean. And so in order to scale this up, we couldn't have 100, robots out in the ocean. So instead, we went back to using that satellite data that is updated every day and matching up the environmental conditions measured from satellites to, we had hundreds of thousands, actually closer to millions of detections of these sharks from those moored receivers in the Delaware Bay and coastal ocean over a 5-year time period. And using a very large data set, we're looking for habitat selection for all sorts of environmental variables, temperature, all sorts of indicators of water clarity and how productive the water was. We fed that in bathymetric features, so their association with different water depths. We fed that into a model, and what you're seeing here is the output of this predictive model, where I can feed it any sort of time period of environmental data, and it'll tell me the probability that sand tigers are likely to be there in that area. And so we did these studies and we're like, this is great. We're moving towards this tool where managers can use or output a more dynamic way of how we're managing when and where these sharks are. And the output was that this information did go into designating the Delaware Bay as essential fish habitat for sand tigers and a habitat area of particular concern. But the output was just this map where we drew some boxes and said, this is just, this is the habitat that we're gonna protect for these species. So it's still good, but we have this great dynamic model that is not being used at the moment. One of the other really useful things that that came out of this project was the fact that the model outputs or the model association, so the habitat selection, was different by different life history stages of those sharks. So we had hundreds of tagged sharks that we were looking at, which included juveniles, mature males, and mature females. And so I was able to build separate models for each of those classes. And what's going to show here is just the prediction of whether or not juveniles are in green, mature sharks are in orangey, and then mature and juvenile sharks where they overlap is in this kind of purpley color. And so the main thing that I want you to notice is that there are times of the year where only juveniles are found, or there's certain times of the year where only mature adults are found, or maybe there's an overlap. And this is really important when we're thinking about managing particular subpopulations within a species. So maybe we feel it's really important to protect nursery grounds for juveniles. It's important to know that there's certain times of the year where they're more likely to be there. Or maybe we're really interested in mature females because we know that's really important for the future success of the population. And so we're able to kind of disentangle some of those nuances in modeling efforts like this. So I've talked a lot about sand tiger occurrence in the mid-Atlantic area, but one thing I want to note is that they're highly migratory and their migration is pretty complex. So this is just from other colleagues' work that shows just the migration of juvenile sand tigers along the whole East Coast. So you can see they range all the way up from New England down into Florida with general summer areas and winter areas. Adding further complexity to this is that some satellite tagging data done by a colleague found that there's actually sexual segregation in their migration patterns. So in the fall and winter, when most of the males and some females and juveniles headed south, as we would expect, there was a whole cohort of adult females that just went offshore, and we have no idea what they were doing out there. And adding to this complexity is that this shark species is really interesting in that they form these really amazing and beautiful aggregations, and we don't really know why they're doing this. It kind of doesn't make sense for a predator unless there's something more going on. And so we were really curious. We knew when we were tagging them in the Delaware Bay that they were in these aggregations, and then we knew this was taken from North Carolina, where they're a very popular tourist attraction for scuba divers. But we didn't know, were they always in these big groups in these schools, or were there certain times of the year where they were kind of splintering off? And this led me into this time where I was really thinking about, well, why do animals even aggregate at all? So there are different external and internal drivers that an animal is experiencing at any given time that influence their movement and in turn change their group size and composition. And this maybe makes more sense if we go back onto land and think about another animal that we consider pretty intelligent. So I'm going to give this example for elephants. So elephants, we can think about, may come together at certain times of the year to defend against predators, right? They form these groups where they feel more protected from predation. Other times they may be sharing knowledge. So a mature female may be sharing information about a really reliable water hole for the rest of her group that she's with. Certain times of the year, then we have sexual segregation. So we know oftentimes mothers and calves will kind of go off and form their own little groups to protect, share in the responsibilities of raising the young, but then also protect the young from more aggressive males. But when there's optimal habitat and food resources are plenty and there's, the spring rains just come, you can imagine a situation where everybody's coming together, everybody's happy and fulfilled. And so you have kind of this mixed population. And then other times of the year where maybe food resources are sparse or there's competition for mating, where you might have these elephants then again kind of splinter off to avoid each other or reduce competition for food. So we can imagine those situations on land. Now let's go back to the ocean. How do you even begin to measure those sorts of things with an animal that's really hard to observe underwater? So it's really difficult, but we're trying. And one of the ways we're doing that is with another type of tag called a VMT, or a Vemco mobile transceiver. So a transceiver, this is what the tag looks like, is up here in the top right. So it's basically a transmitter and a receiver in one. And so similar to how these tags worked, how I introduced how they worked before, you have an animal that swims near a receiver and it can transmit its signal to the receiver. But then what's unique about VMTs is it's basically, how we integrated the receiver technology onto a glider. Here we're integrating the receiver technology into a shark so that the shark now becomes the mobile telemetry platform. One thing that we got to remember is that these tags are still archival. So if you put one of these transceivers inside of a shark, you have to be able to get that shark back to get the data off of it. So that was the challenge. So I actually had the opportunity to work with vets from the Georgia Aquarium to develop this tag implantation technique where we were, this is the kind of the setup for how we would always implant the tags in the sharks, but we had to design a way where we actually internally sutured the tag to the body wall of the shark, and then we sewed it all up so that we knew if we let that shark go and it completed its annual migration, potentially as far away to Florida, and then we're relying on the fact that about 70% of them will return every year. So we're just hoping that it comes back the next year. And then we fished really for that. We tracked them down and we were just trying to convince them to take the bait again so that we could recapture them and get that tag back, and that it would still be where we put it in the shark. So we put 20 of those tags out. There were 12 females and 8 males that we tagged. These were mature sharks only. It was a pretty small scale study, and we got 2 back, which is 10%, which is pretty good. Yeah, so we got 2 back. They were about the same size. They were actually tagged on the same day. We caught them a couple weeks apart the following year. You can see here, this is the healed implantation scar in the bottom left-hand corner there. That's what we used to kind of identify where the tag was likely to be. We opened them back up, got the tags back out, sutured them back up, and sent them back on their way. We weren't really sure what we were going to get. You know, this was the first time anybody had put this type of technology inside of an animal, but we were really thrilled when we got tens of thousands of detections of other tagged animals from 7 and 5 different species from both of those individuals. So what I then did was looked at kind of the social network, if you will, for these animals throughout their year. And that's what is being shown here. These are social network graphs. I'm going to break down what these graphs are actually showing you with a little example from September here. So all of the dots are individual sharks. They're colored by red for female, blue for male. The 2 individuals in the middle, ST1 and ST2, those are the 2 males that were carrying these tags around. So on the left-hand side, these are all individuals that were only detected by ST1 during the month of September. On the right-hand side, you have only sharks that were detected by ST2 in September, and then in the middle is any individuals that were detected by both of them. And then it might be hard to see here, but sometimes there's a black line connecting the 2 black dots, and that means that they detected each other. So I'm going to go through kind of this year and explain to you kind of what we found in a little bit of a cartoony example. So in the summer, the summer and the early fall, we have what we were calling the summering phase. This is when the sharks were in a mixed group in the Delaware Bay and the surrounding coastal ocean. They had a mixed population. You can see there's a lot of red and blue dots, and they're also juveniles and mature individuals represented there. There's a high detection overlap, so there's a lot of individuals that were detected by both of those 2 sharks. And this is really what we're considering a community hotspot. We kind of already knew this. This is where we had been doing our field work. Where, you know, it's probably good habitat for them over the summertime. There's probably a lot of food around. Everybody's kind of together and happy. Then in the fall, they enter this migrating south phase. And this is where previous work had shown indications of sexual segregation, and we think we kind of picked up on this in this work too, where the network graphs switched to, there were a lot fewer females. So there's a lot more blue dots here, a lot fewer red dots here. And remember, these 2 tags were being carried by males. So it seems like we're seeing evidence of that sexual segregation where the group shifted to being more males. They migrated south and they end up off of the North Carolina area. And then they enter what we're calling the bottleneck. So this is where we had a mixed population again. It's a little bit hard to see. There's still a lot of males around, but there were more females again in that area, a lot of juveniles in the area too. So very high community overlap. There's these 2 individuals are basically only detecting similar sharks again. And what we really think is happening here is there's a little bit of a bottleneck, a little bit of a constriction. So sand tigers don't go off super deep. They're constricted by shallow waters out to about 200 meters. And so you can see when they from the depth here that I'm showing you, when they come around that North Carolina area, the habitat is really constricted near shore. There's also the warm waters from the Gulf Stream that are following that shelf ridge there that constrict them in, that they don't enter in. And then there's also, this is the graveyard of the Atlantic, or there's a ton of shipwrecks here. So there's also these sharks are also known to be attracted to shipwrecks in this area, and so that could also be a concentrating feature. And I do have, we'll see if this video plays for you guys. It's gonna be a little bit laggy until it catches up, but you can see here from this video from an underwater scuba diver actually how close these sand tigers are associating. And I just think it's really amazing to think of a top predator and a shark kind of hanging out in these big schools like this. So after this, they enter what I'm calling the dispersal phase. And this is where, again, maybe we're seeing evidence of sexual segregation. So this is the plot from April. You can see there's a lot fewer dots here. Most of them are blue, and actually ST2 didn't encounter any other tagged sand tigers during that time. It did detect other species, but it didn't detect any other species, which is really interesting because the rest of the year it seemed like they were always in these big schools. This could be a time of year where maybe they're limiting interspecific competition for food. Maybe food resources are lower, and so they were spreading out. Maybe they're looking for mates, and the females are all running away and hiding, and the males are all, avoiding each other. We don't really know. We can't quite answer those questions yet, but it's really interesting to start thinking about what they might be doing. What time do I have till?
7:30.
Okay, perfect.
Or 7:20.
Perfect. Okay.
So your plane leaves?
Yeah. All right. So I think this just further, supports the fact that it's really interesting about all of these different nuances that these subpopulations are going through. And this, again, speaks to the point where we need to really, when we're creating these predictive models for these species to inform management, we can also go, a step beyond and also think about the differences within that population of what different portions of that population are doing and how their habitat selection might be slightly different. For example, this is a pregnant female, and again, we might think that for the future success of this population, maybe we really just need to concentrate on what the mature females are doing and focus our efforts, limited conservation efforts, on that portion of the population. And another really interesting thing that I want to mention that's really cool about using these types of tags is that we were not only able to see the differences in the kind of social network for different sand tigers, but these receivers are also compatible with tags that were being put out by other researchers on a number of different species. So this is just kind of the annual snapshot of the interspecific, so the between species social network for these sharks. And so you can see here, for example, ST1, all of these green dots and lines are actually for Atlantic sturgeon. Atlantic sturgeon, we were also studying very heavily in the mid-Atlantic area, and they were recently listed as endangered, and they haven't quite decided what they're going to do with that endangered designation yet, what kind of protection they're going to afford sturgeon. But it's interesting to think about if there's a lot of overlap. Atlantic sturgeon might be serving as kind of an umbrella species for sand tigers. So some of the protection measures that are put in to protect sturgeon may also have kind of carryover effects on sand tiger sharks. And which is an unintended but a really useful benefit. So I have one last project to talk to you guys about that is related to my research, and then I'm going to give just a little bit of a summary of what goes on at the hatchery so that you guys can preface your field trip this weekend. But I'm going to transport you from the Atlantic Ocean back over to the Pacific, and then down actually to the Eastern Tropical Pacific off of Costa Rica, where I've been tagging other species for the last couple of years. And I like to use this photo because whenever I tell people I do research in Costa Rica, they're always like, oh, that must be so great. You get to go out. But a lot of times the days are like this. And so I just want to say it has been a wonderful project to work on, but sometimes you're working in conditions like this. So the goals of this project are really to disentangle the oceanographic and climate impacts on the occurrence and distribution of two really important species in the Eastern Tropical Pacific, the blue marlin and sailfish. The really vibrant ocean ecosystem in this part of the world and amazing biodiversity supports huge industries, commercial fishing industries, recreational fishing industries, as well as conservation and ecotourism industries like whale watching and other types of ecotourism watching type industries. And that's leading to a little bit of conflicts where people are pointing fingers as to who's actually to blame for any kind of disturbances in the population. And then on top of that, we also have climate shifts and strong impacts from the ENSO cycle, so the El Nino and the La Nina cycle, really impacts the oceanography off of this region. And so our goal was to start to try to disentangle what's really going on down there. And to do this, we needed really to have a better understanding of where these species were going. So we used a different type of tagging technology, which is a little bit covered, but we're using pop-off satellite archival tags, which use geolocation. So these are slightly different than those acoustic tags in that they are tags that we deploy on the outside of the animal. They're collecting information, and then at a predetermined time, the tag will pop off of the animal, float up to the surface, and transmit all of that collected data to satellites, which we then get. So geolocation works sunset and calculating data and integrating water temperature and local bathymetry, and then magic, and then you get an estimate of where the animal was. It still feels like magic to me. So that's how we were tracking these animals. And in addition to the tags that myself and my collaborators, this is work that I did while I was at Stanford University. So we deployed tags in 2019 to 2021. But in addition to those tags, we also worked with a number of collaborating institutions who had tagged these species going as far back to the early 2000s. And in this way, we were not only able to expand the number of tags, so the number of observations that we had for these species, but getting observations at different times of the year, but then also integrating different environmental conditions. So here I have now those years colored by the ENSO phase. So red for El Nino, blue for La Nina years. And then in addition to expanding our temporal coverage by working with these collaborators, we were also able to expand our spatial coverage. So then we had tags on fish that were deployed off of Guatemala, Costa Rica, and Panama. Sorry that went, but here's what you really want to see anyways. Hopefully it'll play for you. It looks jumpy on my side too. Oh, technology. Let's see, we'll go back.
It's magic.
Yeah, it is magic. All right, so hopefully this will pick back up. So here's the tag results, which is really cool to see. So these animations go back to the early 2000s. So we almost have 20 years of tagging data for these two species from this part of the world. It was really interesting to know. I think a lot of people might expect that the behavior of blue marlin compared to sailfish might be pretty different. But when we just look at the numbers, it's pretty similar, at least for these populations in this part of the world. So their total draft distance, we were usually observing them for about 2,000 kilometers, and then their daily displacement, so how far they were traveling on any given day, was about 27 kilometers per day, 27 to 29 kilometers per day. What's really interesting, and what was really interesting to us, is that just how resident these fish are to this little area of the ocean. I think when we think of these species, we think of them as kind of traveling across oceans. But here, you know, they are making large movements, but they're pretty retentive in this Eastern Tropical Pacific area, except for one that actually made a little excursion. So out of 20 years of tagging, we only observed one blue marlin make kind of this oceanic excursion out to the west, traveled 6,500 kilometers over about 3 months. Hopefully, there you go. There he goes. So what we're able to look at from integrating all this data is seasonal shifts in space use. I'm going to go through this kind of quickly, but we're able to show, in general, that they are a little bit further south and inshore in the warm season, and then they're pushed offshore and a little bit north during the wet season. We also looked at where their distributions, how their distribution varied over the different ENSO phases. So we're able to show that during the warm El Nino periods, they're shifted a little bit further south and inshore. And then during La Nina periods, especially for blue marlin, they're shifted offshore into the north. So this is really informative in helping the managers understand that potentially some of the differences in fishing success that they're hearing, from commercial and recreational fishers may be due to shifts in the distribution, in addition to possible changes in their population size. This also has implications for how these fish are managed. So this plot here has all of those tracks, and now I have the tracks colored by the different exclusive economic zones. So this part of the world has a lot of little countries all packed together. Some of them have quite expansive exclusive economic zones. But really these fish aren't staying, a fish that I tag in Costa Rica isn't staying in Costa Rica. And so if they really want to manage those populations, they're going to have to work together. And this work is helping to really, helping managers go back to their governments and say, listen, we're really going to have to come together and make sure that we're all managing these species in a way that we're all happy with, because we all need this resource, but it's really what's happening in our national waters that is going to be having an effect on these species. Because one of the major results of this work is that these two species are only spending 7 and 8% in areas beyond national jurisdiction. So they can't just blame, whatever's happening to the populations on whatever's happening in unregulated international waters. Really, they can make a difference on how these populations are managed within national waters, but they have to cooperate. So I've shown you how, the basics of dynamic ocean management and how we integrate different observations of the biological observations of where animals are with the environmental conditions. And I've showed you how we can create tools to, potentially inform management decisions, but the outcome is we're still not quite there yet. So the case study that I presented to you in the beginning of the California drift gillnet, you may be aware that the state has decided we're just buying out the rest of those permits. And I'm not saying that that's not a good choice. Drift gillnets and gillnets in general are a pretty problematic fishing style. It's really hard to control what gets caught in a drift gillnet. So that fishery will, no longer be happening in California waters. And some good has come of that. We've really pushed, the fishery to move towards other fishing styles, which will be a little bit more selective. So deep set buoy gear has shown promising, and this kind of movement to change the type of gear that these fishers are using really came out of all of the protective measures that were being placed on fishers. But we still have the opportunity to rethink how we're managing our oceans using all of this amazing technology and all these observations that we have now. And so I like to leave people with progress isn't possible without change. And I think, there's a lot of bureaucracy. Our management systems just aren't set up right now to kind of be able to integrate and adapt on the type of dynamic scale that scientists are able to produce, with all of these models. And so we kind of need to work together with people who are good at computers and good at thinking through strategically, and then the political system, and rethink how can we start to use some of this technology to actually make meaningful change in how we're managing and conserving our resources. And I think I have a little bit more time, so I wanted to give you tie this into a little bit of some of the work that you guys are going to be seeing in your field trip on Saturday for you, those of you who are able to go. And what I really want to highlight is kind of another type of way that we can look towards solutions for a more sustainable relationship with our ocean resources, which I think is really exemplified by our Sustainable Seafood Program. That program has a couple different arms of research. the fisheries replenishment, kind of farm to table. So thinking about how we might be able to farm or aquaculture different types of products and bringing them to table. And then integrated multi-trophic aquaculture, which integrates bivalves, invertebrates, and seaweeds into the system to try to address some concerns about water quality, but then also producing additional products in addition to the fish. So you'll hear a lot more about this, but what I wanted to give you was just a little bit of an overview of the fisheries replenishment side of things. And we're going to focus on the California white sea bass, which is the fish that you'll see. So for context, the white sea bass in California waters, this is the time series of catch landings in the commercial gill net fishery. So this was a gill net fishery. This time series goes back to 1936. So you can see there's some natural variability in the fish landings with some booms and some busts. But then around the early 80s, really the fishery had collapsed. In response to that, there was a program that was developed, and in 1986, the first experimental release of more than 2,000 juvenile white sea bass took place in Mission Bay, and these were fish that were raised at the property just right across the street. And in addition, there was several more years of the fishery still wasn't rebounding. So in 1994, there was an implementation to ban nearshore gill net fishing by California State Proposition 132. In 1995, the first marine hatchery became operational. This is where you guys are going to be going on Saturday. So this was built on land donated by San Diego Gas and Electric Company on the Agua Hedionda Lagoon in Carlsbad, California. So this is a joint program between the California Department of Fish and Wildlife and Hubbs. In 1999, just so you get a little time history of the program. So the first legal sized tagged hatchery raised white sea bass was recaptured in 1999. In 2001, that was the first year that we had really ramped up operations and we were capable of producing 100,000 white sea bass that were released into Southern California waters. All of that work, by 2004, we'd released 1 million white sea bass into California waters, and by 2014 we'd released 2 million. So that was when we reached 2 million white sea bass released. So this is really this program that falls under the fisheries replenishment side of our sustainable seafood program is the Ocean Resources Enhancement and Hatchery Program. So that's the program through California Department of Fish and Wildlife. It's funded actually by sales of stamps that are sold on commercial and sport fishing licenses, and then also some funding from the Federal Sport Fish Restoration Act. And so in order to get to the point where we were able to produce 100,000 fish per year, you can't just rely on one spawning season. So white sea bass are a spring spawner. And so what we had to do in the early days was a lot of work trying to figure out what are the environmental conditions that we need to have different tanks in order to spread spawning out throughout the year so that we can get to producing 100,000 fish. And so this just shows, what we have now is we'll have tanks that you'll see with different brood stock in them, different groups of male and female mature fish. And then what we're doing is manipulating the conditions so that at any given time of the year, we always have a tank that thinks they're going into spring. And so here we have the water temperature shown in blue and the light levels shown in yellow, and then spawning events are these red lines. And so you can see as we bring tanks up, we increase day length, and we bring the water temperatures up, then we'll trigger these spawning events that we can now have happening all year round. And white sea bass culture, just to run you through it, and you'll get to see a lot of this. Summer's a good time to visit the hatchery. So we have a spawn, and then we will have the eggs, which we can then separate out of the tank. We are able to sort them for fertilized eggs and non-fertilized eggs. We look at egg quality and we think about the factors that influence egg quality. And we're able to do estimates of the number of eggs, and that helps us inform how many females contributed to that spawn. And we do a lot of work of tracking the genetics of the fish to make sure that we're, varying and keeping a diverse genetics going. So after about 2 days, the eggs will hatch into larvae. or into embryos, sorry. So you'll start to see them kind of form these embryos where they're still feeding on the oil sac that they're born with. So, you know, they don't have mouths yet, so they're absorbing that nutrition. After about 10 days, then they'll hatch out into larvae. And that's when, there they go. And we'll move them into different tanks throughout, you know, depending on what stage they're in. And you'll see some of that happening during the field trip. So then they go into this nursery stage where we grow them from 10 days to about 100 days post-hatch. Once their mouths form, I'm sorry, this is jumpy. Once their mouths form, we actually start them out on live feeds where we feed them little brine shrimp. And then as they get bigger and bigger, we'll slowly work them over to dry feeds or these pelleted fish foods. Once they get to a certain size, we're able to tag them with coated wire tags. This is how we track whether or not fish that are recaptured in the wild were from our hatchery. And so this is a little machine where we're implanting this, what you can see here, that's actually the tag. So it's very, very small, about the size of pencil lead. And on each of the tags is a unique serial number, which allows us to track that fish back to what tank, what spawning event produced that fish. So those tags are implanted into the cheek muscle of the fish where it will hopefully carry it for the rest of its life. The fish then move into a grow out stage. So the juveniles are moved into they were transitioned from recirculating aquaculture systems where the water is very clean and UV sterilized and filtered. We move them into raceways where they start to experience wild water. So we'll filter the water, but we're no longer sterilizing it, so they can start to get exposure to natural things that they would be exposed to out in the wild. And then they're eventually transitioned into outdoor net pens, which most of these are maintained and the fish are cared for by volunteer fishers in a variety of different fishing nearshore port areas along Southern California, where they stay until they get to a releasable size. And so fish are released at about 8 inches. And so far we've released 2.7 million, approximately, white sea bass into Southern California, trying to enhance the wild populations. So that's a little teaser about some of the work, and another solution that we're working on at Hubbs related to fish. That's all I had for you today. And so I thank you for your attention and your time, and I would be happy to take any questions. Yeah.
And you probably want to repeat the questions just so
Okay.
We have a double the size, well maybe not quite. We have 10 people on so you got a good audience tonight.
Great.
What are the sand sharks eat?
Sand tiger sharks, so they have you can tell a lot about sharks and what they eat based on what their teeth look like. Right. Sand tiger sharks are very commonly in aquariums because they're pretty docile, but they look very scary because they have these kind of pincer, skinny, sharp teeth that actually are useful for catching slimy little small things. Like we catch them using menhaden, or it's called bunker, just a little kind of forage fish that is common in that area. They also are known to eat things like squid, crabs, things they can catch, that sort of thing. So, you know, not big mammals, their teeth aren't meant for that. Little small fish.
Go ahead.
Are the fishermen fairly cooperative with you when they announce not to go to certain areas? No.
It depends. Yeah, I mean, so it depends where in the world you are.
Yeah.
That often relates to what enforcement looks like. I will say we're getting, in general, I think, yes, especially, you know, commercial fishermen don't want their fishery closed down, and we're getting better and better at tracking where boats are. So, you know, not only are a lot of boats required to have certain tracking technologies on them, but also we're getting better at just using satellite data and true color imagery to spot where fishers are in the water. And that's a growing movement. that is an interesting thing to think about in terms of privacy, but that's not for us to debate here. But yeah, I would say in general, I mean, you can often see even all over the world in certain areas where a marine protected area will be established, the fishing pressure, you know, from one year to the next, you'll see will usually just concentrate right on the edges of those boundaries. You can almost identify boundaries that we placed in the ocean by just looking at where fishing effort is. So I would say in general, sometimes they might not agree with the reasons behind those designations, but in general, you know, yeah, well, yeah. Yeah.
An online question, maybe
Yeah.
we can alternate back and forth. So George wants to know, are you aware of multi-species tracking and interaction studies taking place on the West Coast?
Yeah, so I'm not aware of if he's talking about a particular study, but there's a lot of work going into trying to see integrating instead of just focusing on one single species, trying to integrate where things are overlapping. One of the tricky things that's important to remember is that, like, especially for satellite tags, the error locations around all of those positions is usually quite large. So for the tracks that I showed you for those marlin and sailfish, those are just estimates, and actually the error estimates around any one of those points, it could be up to 50 kilometers. And so when you see kind of two tracks cross, that doesn't mean that we have very high confidence that those two individuals actually encountered each other. Different technologies will allow you, so like acoustic telemetry, your detection range is much finer resolution. So those plots that I showed you about the sand tigers being together, they were within 1 kilometer of each other. That's still quite big, but it's better than tens of kilometers. And then there are some other tags where they can actually detect like predation events, which will help you get at predator-prey relationships. And so it's tough when it depends on what the question is of what is the multi-species interaction. Are you just looking for broad habitat and distribution overlaps, or are you thinking more about like the actual interactions between predators? And it just depends whether or not we have the technology to get there.
Haley wants to know, how do you know when this is successful? In other words, when the white sea bass does not overpopulate.
Yeah.
How will we know that we've gotten back to the numbers that you would love to get back to?
That's a great question. I think we've seen some recovery in the population. Part of that, I think, is due to some of those bans on different fishing that I mentioned were implemented. And then I would like to think that the replenishment program has made an impact. We're currently looking at recovery rates of those coated wire tags, so seeing how often fishermen are recovering hatchery raised fish. And then we're also very new to the program is integrating genetics and using genetic techniques to look at how related wild populations or fish that are caught in the wild might be to the fish that we have as broodstock, to looking at like what is the contribution that we're actually making to the wild population. I would say that those types of things are still in the works right now, but those are the types of things that we're looking at to see what is, how much of an impact are we making? Because I think we don't want to get to a point where it's like 80% are hatchery raised fish. We're hoping that we're enhancing the wild population, that then there's wild spawning of those fish happening that then helps the fishery rebound.
What's the reproductive strategy for your sand shark, your sand tiger shark? Are they live birth, they eggs? And
yeah,
so where are they given? Where's the nursery?
I love that you asked this question because they're such an interesting species. They're my favorite species of probably marine animal. So sand tiger sharks have live birth. So all sharks have 2 uteruses. And sand tiger sharks have this really unique and interesting thing where they have intrauterine cannibalism. So the first embryo and fetus to develop in each of those uteruses will go around and eat and consume all of its other brothers and sisters until it is the only one left. And so a sand tiger shark will give birth to 2 pretty well developed, up to 1 meter long when they're born, but only 2. individuals per female, and we think they are reproducing or every other year, so on a 2-year reproductive cycle. And I think the estimate is that females are only reproductively active for about 20 years, so that's 20 years divided by 2,
Yeah, 20
yeah,
animals.
not very many.
Yeah, 20 animals. Well, it's
Yeah.
a replacement. It's enough of a replacement.
Yeah.
And is there a identified nursery area that you're going to see
Um,
higher concentrations of those
yeah, it's
newborns?
it's usually some of those coastal embayments. I would say I don't know that we have it quite nailed down like exactly where they're born, but we will get young of the year, so freshly, they still have belly buttons up in the Delaware Bay, but a lot of those young of the year will also keep going up into New England, into waters that the mature individuals don't. So kind of up in those New England estuaries, I would consider a nursery area for those juveniles. But then a lot of the shallow kind of embayments along the whole East Coast at any given time of the year probably has some juveniles.
Okay.
Yeah.
One in the back.
What are the next steps that are needed for like sort of the MPA development, like off the Cocos Islands and in that area in Costa Rica?
Can you repeat the question?
Yeah, so the question was about the MPA developments in the Eastern Tropical Pacific, mentioning the Cocos Island, which recently had an expansion of their marine protected area. I would say the newest development probably might be of interest is they're looking at kind of habitat connectivity corridors between these protected areas on these different islands in that part of the world. So they have a lot of those marine protected areas around those islands, they've expanded to try to push out mostly the international tuna purse seiners that are coming through. But then what they're looking at too are seasonal migratory corridors to help connect these different protected areas. So yeah, so I don't know if that corridor protected area has officially been implemented yet, but it's very close if it hasn't. But I would say that's kind of the newest development, is there's some expansion going on and then connectivity between those areas.
How does one determine that one of your fish are tagged? If I catch it, how do I know that?
That's a great question.
I
know. So you can't. And so what we ask, so there's a big effort right now to collect fish heads. So at a lot of the different landings, there are freezers and stations set up for fishers to tag, write down their information, put it in a bag, and collect these heads in different freezers. And then we are going around and collecting all of those heads and scanning them for tags, and then also integrating those samples into our genetics works that's going on. So really, the best thing you can do is if you catch a white sea bass of legal size, cut off its head and put it in a bag and find one of these freezers. Yeah, so we have freezers up across at Mission Bay, but then there's also freezers stationed all throughout Southern California, and there's lists of that online that you can find.
Do you find that the tags migrate in the fish?
That's a great question. So yeah, it's a good question that we want to look into. And so that's something that we're also thinking about doing too, is once we do get back fish heads that we can scan for the tag, is running that through an x-ray and trying to figure out, because I think we're recapturing a lower number of fish of legal size than we would expect based on how many that we're putting out. And so there is a question about whether or not those tags are migrating into different parts of the body, or the fish kind of expelling them at some point. But we have had a fish that was recaptured 20 years after it was released. So we do know for some fish that they are retained for a really long time.
Kind of jump in on that, because one of your questions that is on, and we'll go here, but on that tag recovery, what about a predator eating that as a smaller fish? Would the predator retain that or excrete it, or what do you?
It's hard to say. I mean, they're about the size of a piece of pencil lead. I would imagine that it'll probably get excreted, but something that small could also probably, I don't know. And I don't know if you have any.
I don't know that we could address that.
Yeah, I don't know. It would be, such a rare thing, and we're not scanning sea lion bellies for these tags, but it would be interesting.
Most of the rest of us aren't going to recognize it if it gets frustrated.
Well, yeah, you saw how small it was, so it would be nearly impossible, I think, to recover that. But I will say that some of the tags that I use, the electronic tags, we will sometimes be able to document predation events because the temperature will all of a sudden spike up. So something that is in 60 to 70 degree water all of a sudden spikes up to 90, you can say, okay, a worm, a shark, a white shark, or a big tuna, or something that's creating body, or a mammal, something that is warm-blooded ate that, and then you can see that sometimes happening. But
why
these little
end
tags.
with a question from Anne, because we know you're time sensitive, and
Yes,
I
appreciate it.
That. I also want to make sure Anne gets her question.
Okay, back to your sand tigers.
Yes.
What is it about Rex that appeals to that?
Yeah, that's a good question. I think, the East Coast, I don't know how familiar people are, the ocean is pretty boring compared to the West Coast. It's very sandy. There's generally not a lot of structure, not a lot of rocks, habitats happening. I think it's just the structure. I've asked a lot of the divers, what are they doing there? And they're like, they're just hanging out. So it doesn't, I think they've tried to document with cameras whether or not there's high feeding activity at night or something like that. So these structures are not only aggregating sharks, the predators, but they're also aggregating all the bait fish that you would expect. So it's possible maybe it's a local concentration of food. It's possible they just want to hide in a little shipwreck. I'm not really sure what it is. I don't know what.
Has anybody ever instrumented the area of a wreck to see how many things get back from tagged animals?
Yeah, so they more recently, there have been some groups that have been putting receivers there. One thing that's interesting too that I didn't mention is, so I mentioned that females have this every other year, so reproductive cycle. So one summer, mature female will go to the Delaware Bay, and then the next summer, usually she doesn't, and she stays down in North Carolina, and we're thinking off of those wrecks. And so they'll have sand tigers there almost year round, but the populations will shift. And we're thinking that they're hanging back in, it'll get way hotter there, but we think that's when they're pregnant, is when they're taking that summer off, because we've actually had vets bring out ultrasounds out on the boat to try to ultrasound the females that we're catching up in the Delaware Bay, and they've documented pregnancies up there. They've also done sperm quality assessments of the mature males, and it doesn't seem like they're reproductively active when they're in that part of the ocean. But they have documented with underwater ultrasounds pregnant females on the wrecks off of North Carolina. And so we think that there's that going on too. Why are they hanging out down there? They must like the structure, but then also the environment, there's a little bit of selectivity for that. And maybe they're just avoiding everybody else when they're pregnant. It's a good question though. Yeah.
All right, Madam President.
Oh, thank you. I thank you so much for your
thank
presentation.
Real applause.
Yes, and thank you so much for working with us and your schedule. And,
don't worry
coming
about
out
it.
Here.
Yeah.
What I want to do is show everyone at home what the boat looks like, and because we have, oops.
No, it's okay.
Okay,
We
we
don't
have
need anymore.
lots of room, and I don't know if you all can see.
Yeah, it looks good.
We have, lots of room, and it's, been a great evening. And it's been great because you get the actual feedback, the actual reactions, and plus tonight's been great because I've been able to put bodies and people with the names that we see on Zoom. So, it's been a lot of fun this evening, and I'm so glad you all were able to make it. And I'm so glad you all joined us from home as well. Next month, again, we have I just went blank. Jessica Krantz here on the Legacy and via Zoom. If you have any questions or comments, you can email us at acssandiego10@gmail.com or look us up on the internet at acssandiego.org. Saturday, don't forget, is the tour of the Hubbs-SeaWorld Research Institute's fishery in Carlsbad, and that's at 10 a.m. Try to get there a little early for parking so that we can all group and then move around the tour together. Anything else?
Before we go, if you guys want to, let's go into the bow and do a quick group shot of everybody so we can put it in our newsletter if you want. Because if you go in the
Unless
bow.
you're in the witness protection.
All right, I think that's all for Zoom. Thank you all so much again, and have a great evening. Thank you. We're going to do our
we have an
opportunity
opportunity
drawing
drawing.
in just a second.
Okay.
Let's do that.